Good day, class. For today's lecture, we will be discussing about clinical enzymology. Upon the completion of this chapter, the clinical laboratorian, or you, our students, should be able to do the following. Define the term enzyme, including physical composition and structure. Classify enzymes according to the International Union of Biochemistry. Discuss the different factors affecting the rate of an enzymatic reaction. We'll also explain enzyme kinetics, including zero-order and first-order kinetics. Explain why the measurement of serum enzyme levels is clinically useful. Discuss which enzymes are useful in the diagnosis of various disorders, including cardiac, hepatic, bone, and muscle malignancies, and acute pancreatitis. Discussing the tissue sources, diagnostic significance, and assays, including sources of error, for the following enzymes such as creatine kinase, lactate dehydrogenase, aspartate aminotransferase, alanine aminotransferase, alkaline phosphatase, acid phosphatase, gamma glutamyl transferase, amylase, lipase, cholinesterase, and glucose 6-phosphate dehydrogenase. We will also evaluate patient serum enzyme levels in relation to disease states, discuss the clinical importance for detecting macroenzymes, and lastly, discussing the role of enzymes in drug metabolism. But before anything else, class, let's define what are enzymes. Enzymes are specific biologic proteins that catalyze biochemical reactions without altering the equilibrium point of the reaction or being consumed or changed in composition. The other substances in the reaction are converted to products. The catalyzed reactions are frequently specific and essential to physiologic functions such as the hydration of carbon dioxide, nerve conduction, muscle contraction, nutrient degradation, and energy use. Found in all body tissues, enzymes frequently appear in the serum following cellular injury or sometimes in smaller amounts from degraded cells. Certain enzymes, such as those that facilitate coagulation, are specific to plasma and therefore are present in significant concentrations in plasma. It can be either in plasma or serum enzyme levels that are often useful in the diagnosis of particular diseases or physiologic abnormalities. In simpler terms, class, enzymes are proteins that help speed up our metabolism. Or it can also be of use in a chemical reaction inside our bodies, such as our digestive enzymes, so they help digesting the food that we eat, degrading the nutrients from there, extracting the nutrients. They can also build some substances and may also break others down. All living things have enzymes, so our bodies naturally produce these enzymes, class. Once again, enzymes catalyze many specific physiologic reactions, and these reactions are facilitated by the enzyme structure and several other factors. Since enzymes, class, are also considered proteins, so you expect that each enzyme contains a specific amino acid sequence that comprises the primary structure of the enzyme and the resultant polypeptide chains that are seen twisting. That's your secondary structure. What's unique about the secondary structure, class, is that you can see the alpha helices or the beta-plated sheets as seen in the illustration above. The local foldings and twists that you can see in the secondary structure, class, will then becomes a 3D folding pattern, which means that there will be side chain interactions between the other amino acid sequence. And that's why it will look like that in the tertiary structure. And then, if the enzyme contains more than one polypeptide unit, that is what we now call the quaternary structure, which refers to the spatial relationships between the different subunits. Imagine the encircled portion as the enzyme. So you can see there, class, that in an enzyme, there is a regulatory region and also an active site. Each enzyme class contains an active site. This active site is often a water-free cavity where the substance on which the enzyme would react to, or where the enzyme would bind to, or what we call the substrate, interacts with a particular charged amino acid residue. So this active site class looks like a puzzle piece wherein you will put the substrate or the other puzzle piece in order to complete the picture. And whenever the substrate binds to that active site class, that is where chemical reaction happens. Other than the active site, there is also an allosteric site, or otherwise known as the regulatory region. This is a cavity other than the active site. And this area here may bind regulator molecules and thereby be significant to the basic enzyme structure. When regulatory molecules bind to the enzymes class, it can either make or break the function of these enzymes. So it can also alter its function. What do you think are the different factors that could affect enzyme reactions? The first would be enzyme concentration. Because enzymes catalyze physiologic reactions, the enzyme concentration affects the rate of the catalyzed reaction. So this means as long as the substrate concentration exceeds the enzyme concentration, the velocity of the reaction is proportional to the enzyme concentration. Okay, so this means that in simpler terms, increasing enzyme concentration will speed up the reaction as long as there is substrate available to bind to. Remember that an enzyme needs to be binded or bound to a substrate to make a chemical reaction. So once all of the substrate is bound, the reaction will no longer speed up, since there will be nothing for additional enzymes to bind to. Therefore, the higher the enzyme level, the faster the reaction will proceed because more enzyme is present to bind with the substrate present. Is that understood now? Another major influence on enzymatic reactions is substrate concentration. In 1913, Michaelis and Menten hypothesized the role of substrate concentration in the formation of the enzyme-substrate complex. According to their hypothesis class, the substrate can readily bind to a free enzyme at a low substrate concentration. With the amount of enzyme exceeding the amount of substrate, the reaction rate steadily increases as more substrate is added. In other words, increasing the substrate concentration will also increase the rate of reaction to a certain point. Why to a certain point, miss? It's because we're only adding substrates and the readily available free enzymes are already bound to the previously added substrates. So even if you keep on adding any substrate, there will be no increase in the rate of reaction. Again, adding more substrate will have no effect on the reaction as the available enzymes will be saturated and working at their maximum rate. The third factor that could affect enzymatic reactions are cofactors. These are non-protein entities that must bind to particular enzymes before a reaction occurs. Common activators or inorganic cofactors are metallic. An example would be calcium, iron, magnesium, manganese, zinc, and potassium. Write that down. Again, calcium, iron, magnesium, manganese, zinc, and potassium. Those are metallic activators. The non-metallic activators class are bromine and chloride. Again, bromine and chloride are non-metallic activators. The activator may be essential for the reaction or may only enhance the reaction rate in proportion with concentration to the point at which the excess activator begins to inhibit the reaction. Activators class, they function by alternating the spatial configuration of the enzyme for proper substrate binding, linking substrate to the enzyme or coenzyme, or undergoing oxidation or reduction. Some common coenzymes or organic cofactors, remember class that activators are inorganic cofactors, while the coenzymes are organic cofactors. These are nucleotide phosphates and vitamins. Coenzymes serve as second substrates for enzymatic reactions. When they are bound tightly to the enzyme, coenzymes are called prosthetic groups. For example, the nicotinamide adenine dinucleotide, it's a cofactor that may reduce to nicotinamide adenine dinucleotide phosphate in a reaction in which the primary substrate will be oxidized. Increasing the coenzyme concentration class will also increase the velocity of an enzymatic reaction in a manner synonymous with increasing substrate concentration. Therefore, that's the reason why it's called second substrate. When quantitating an enzyme that requires a particular cofactor, that cofactor should always be provided in excess so that the extent of the reaction does not depend on the concentration of the cofactor. So to go back, when the coenzyme class is already bound to the enzyme itself, you call it prosthetic group. So since now it is already called a prosthetic group, the enzyme portion is called apoenzyme, and its respective coenzyme, when they form a complete and an active system, you call it holoenzyme. There are also other enzymes class, mostly digestive enzymes. They are originally secreted from the organ of production in a structurally inactive form. This means they are not active. And when they are in this inactive form, they are called proenzyme or zymogen. So once they are already put to use, these other enzymes later alter the structure of the proenzyme. Remember, the proenzyme is the inactive form of the enzyme to make active sites available by hydrolyzing specific amino acid residues. And this mechanism class of inactivating certain enzymes prevents digestive enzymes, remember, our digestive enzymes, from digesting their place of synthesis or from digesting the organs or the tissue surrounding it. The fourth factor that could affect enzymatic reactions would be inhibitors. Reactions class may not progress normally if a particular substance, such as inhibitors, interfere with the reaction. There are actually three different types of inhibitors: competitive, non-competitive, and uncompetitive inhibition. For competitive inhibition class, these inhibitors physically bind to the active site of an enzyme and they compete with the substrate for the active site. With the substrate concentration significantly higher than the concentration of the inhibitor, the inhibition is reversible because the substrate is more likely than the inhibitor to bind the active site and the enzyme has not been destroyed. Kindly take a look at the picture that is shown to you in the screen. You can see there that there's an enzyme whose active site is triangular in shape, and The substrate also has a triangular shaped base. So once they connect, they create an enzyme substrate complex and therefore produce a certain product. But in competitive inhibition class, you can see there that the enzyme still has the same shape as the active site, so that's still triangular in shape, but the inhibitor is being represented by a triangular shaped base, but it has a box or a square as its head. So when they create an enzyme inhibitor complex, then the reaction would not proceed. This is also another illustration on what competitive inhibition is about. Another type of inhibitor would be non-competitive inhibition. This binds an enzyme at a place other than the active site and may be reversible in that some naturally present metabolic substances combine reversibly with certain enzymes. Non-competitive inhibition also may be irreversible if the inhibitor destroys part of the enzyme involved in catalytic activity. Because the inhibitor binds the enzyme independently from the substrate, increasing substrate concentration does not reverse the inhibition. So as you can see in the illustration above class, when the inhibitor binds to the enzyme, there are some changes that is happening in the active site. Therefore, the enzyme and the substrate complex can still be done, and at the same time, products will not be generated. And the last type of inhibition would be uncompetitive. This is also another kind of inhibition in which the inhibitor binds to the enzyme substrate complex. So increasing substrate concentration results in more enzyme substrate complexes to which the inhibitor binds and thereby increases the inhibition. The enzyme substrate inhibitor complex does not yield any product. Okay, brief recap. The normal reaction for enzyme and substrate is found in the first picture. You can see there that the substrate really fits the active site of the enzyme. For the competitive inhibition class, the inhibitor will compete with the substrate, so more or less they have the same shape as the active site of the enzyme, and so that's why there is an instance that the substrate could no longer bind to the enzyme. The solution for competitive inhibition is to increase substrate concentration so that the inhibition will be reversible. For non-competitive inhibition class, the inhibitor will bind to the enzyme other than its active site. So probably it's allosteric or regulatory site. And in doing so, the active site of the enzyme would no longer be the same because it was already altered. So this means even if you add excess substrate, there will be no changes, no irreversible inhibitory action. And for uncompetitive inhibition, the inhibitor would bind itself to the enzyme substrate complex. If you add excess substrate, then there will be more enzyme substrate complex. Therefore, that will also increase the inhibition. Another factor that could affect enzymatic reactions are isoenzymes, or otherwise known as isozymes or isoforms. The rationale about this class is that even though a particular enzyme maintains the same catalytic function throughout the entire body, that enzyme may exist in different forms within the same individual. The different forms may be differentiated from each other based on certain physical properties such as electrophoretic mobility, solubility, or resistance to inactivation. The term isoenzyme is generally used when discussing such enzymes. However, the International Union of Biochemistry suggests restricting this term to multiple forms of genetic origin. An isoform, on the other hand, results when an enzyme is subject to post-translational modifications. So this means that isoenzymes and isoforms contribute to heterogeneity in properties and function of enzymes. In simpler terms, class, some enzymes, let's say for example creatine kinase, it has been subdivided into 3 other isoenzymes basing from its tissue distribution. So the CKMM, majority of it comes from the muscles. The CKMB, majority of it is found in the heart, while the CKBB, majority of it is found in the brain tissue. So meaning class, when testing these in the laboratory, it will give us an information as to what part of the body of this patient has a problem. Aside from creatine kinase class, we also have lactate dehydrogenase, amylase, and also aldolase, and some other enzymes that also has isoenzymes in them. Another factor would be temperature. When increasing the temperature, this also usually increases the rate of a chemical reaction by increasing the movement of molecules and the rate at which intermolecular collisions occur, and lastly, the energy available for the reaction. This is the case with enzymatic reactions until the temperature is too high enough to denature the protein composition of the enzyme. For each 10-degree increase in temperature, the rate of reaction will approximately double until, of course, the protein is denatured. Each enzyme functions optimally at a particular temperature, which is influenced by other reaction variables, especially the total time for the reaction. The optimal temperature is usually close to that of the physiologic environment of the enzyme. However, some denaturation may occur at the human physiologic temperature of 37 degrees Celsius. The rate of denaturation increases as the temperature increases and is usually significant at 40 degrees Celsius to 50 degrees Celsius. Because low temperatures render enzymes reversibly inactive, many serum or plasma specimens for enzyme measurement are refrigerated or frozen, that is to prevent activity loss until analysis. So in the laboratory class, after we extract the patient sample, we centrifuge it, we get the serum or the plasma of the patient, and then refrigerate it or frozen it. Storage procedures may vary from enzyme to enzyme because of individual stability characteristics. If you repeat freeze and thaw, or repeated freezing and thawing, however, tends to denature protein and should be avoided. Because of their temperature sensitivity, enzymes should be analyzed under strictly controlled temperature conditions. Incubation temperature should be accurate within plus minus 0.1 degrees Celsius. Laboratories usually attempt to establish an analysis temperature for routine enzyme measurement of 25 or 30 or 37 degrees Celsius. Attempts to establish a universal temperature for enzyme analysis have been unsuccessful, and therefore, the reference ranges for enzyme levels may vary significantly among laboratories. Another factor that could affect enzymatic reactions would be the way we store our samples for enzymatic analysis. So negative 20 degrees Celsius is the preservation or storage temperature for longer period of time. 2 to 8 degrees Celsius or refrigeration temperature is ideal for substrate and the coenzymes, while room temperature is ideal for storage of lactate dehydrogenase enzyme. That's for LD4 and LD5. By the way, for lactate dehydrogenase, when we get to the enzyme lecture topic already, you will know that lactate dehydrogenase has 5 isoenzymes plus a 6th isoenzyme that is referred to as alcoholic dehydrogenase enzyme. You will know that soon. Next would be pH. So enzymes class are proteins, remember, that carry net molecular charges, meaning the changes in pH or hydrogen ion concentration may denature an enzyme or influence its ionic state, resulting in structural changes or a change in the charge of an amino acid residue in the active site. Hence, each enzyme operates within a specific range and maximally at a specific pH. The most physiologic enzymatic reactions occur in the pH range of 7 to 8, but some enzymes are active in wider pH ranges than others. In the laboratory, the pH for a reaction is carefully controlled at the optimal pH by means of appropriate buffer solutions. An example for this would be acid phosphatase and alkaline phosphatase. By the name itself, this means that you have to maintain the testing area or its reaction at a pH where the enzyme is viable at. Is that clear? So when you say acid phosphatase, then that means you have to maintain the testing or the reaction at an acid pH. For alkaline phosphatase, the same thing. You have to maintain the reaction at an alkaline pH. Another factor would be hemolysis. At all times, class, we always make it sure that we avoid hemolysis or we avoid the disruption or the rupture of the red blood cells during blood collection because hemolysis mostly increases enzyme concentration. This means that if there's a pseudo increase of enzymatic concentration or enzyme concentration rather, that means we will be what? Reporting false results. And that could probably cost the life of the patient due to wrong diagnosis. So it's always best to avoid hemolysis. The last but not the least would be lactescence or milky specimen. This also does the opposite. It decreases enzymatic concentration or enzyme concentration rather. It has been established, class, that lipemia is a significant source of analytical errors in the clinical laboratory settings, and therefore we should remove it prior to measuring biochemical parameters. Let's now proceed to enzyme classification and nomenclature. To standardize enzyme nomenclature, class, the Enzyme Commission of the International Union of Biochemistry adopted a classification system in 1961. The standards were revised in 1972 and 1978. The IUB system assigns a systematic name to each enzyme, defining the substrate acted on, the reaction catalyzed, and possibly the name of any coenzyme involved in the reaction. Because many systematic names are lengthy, a more usable trivial recommended name is also assigned by the IUB system. In addition to naming enzymes, the IUB system identifies each enzyme by an EC numerical code containing 4 digits separated by decimal points. The first digit, class, places the enzyme in its classifications. So, there are 6 classifications: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. Oxidoreductase being number 1 and the ligases would be number 6. The second and third digits represent the subclass to which the enzyme is assigned, and the final and fourth numbers is the serial number that is specific to each enzyme in a subclass. Let's discuss this one by one. For oxidoreductases class, their main function is to catalyze the removal or addition of electrons. This means they do redox reaction. Examples would be cytochrome oxidase, lactate dehydrogenase, malate dehydrogenase, isocitrate dehydrogenase, and glucose 6-phosphate dehydrogenase. Represents number 2 in the enzyme classification would be transferases. They catalyze the transfer of a chemical group other than hydrogen from one substrate to another. Examples would be creatine kinase, aspartate aminotransferase, and alanine aminotransferase, and also ornithine carbamoyl transferase. Representing number 3 in the first digit in the 6th classification of enzymes would be hydrolases. Their main function is to catalyze the hydrolysis or splitting of a bond by the addition of water. For hydrolases class, they do hydrolytic reactions. Example of which would be the esterases, so acid phosphatase, alkaline phosphatase, lipase, also the peptidases, trypsin, pepsin, leucine aminopeptidase, and also glycosidases for amylase and galactosidases. Representing number 4 in the enzyme classification would be the lyases. Their main function is to catalyze the removal of groups from substrates without hydrolysis. The product contains double bonds. Okay, that's for lyases. An example would be glutamate decarboxylase, pyruvate decarboxylase, aldolase, tryptophan decarboxylase. Representing number 5 would be isomerases. Their main function is to catalyze the intramolecular arrangement of the substrate compound. Example of which would be glucose phosphate isomerase and ribose phosphate isomerase. And lastly, for the ligases, they catalyze the joining of two substrate molecules coupled with breaking of the pyrophosphate bond in ATP or similar compound. The next following slides class is the classification of frequently quantitated enzymes. Together with this, you can see what are the enzymes part of each of the 6 classification of enzymes, their recommended name, their standard abbreviation, as well as their enzyme commission number. You have to memorize class the enzyme commission numbers because that will come out in our exam. For lactate dehydrogenase class, EC number would be 1.1.1.27. Glucose 6-phosphate dehydrogenase would be 1.1.1.49, and glutamate dehydrogenase would be 1.4.1.3. Brief recap, the first digit places the enzyme in its classification. So 1 here represents oxidoreductases. The second and third digits again represents the subclass to which the enzyme is assigned. And the final and fourth numbers is a serial number that is specific to each enzyme in a subclass. Under transferases, aspartate aminotransferase would be 2.6.1.1, alanine aminotransferase 2.6.1.2, creatine kinase 2.7.3.2. Gamma-glutamyl transferase 2.3.2.2, glutathione S transferase 2.5.1.18, glycogen phosphorylase 2.4.1.1, pyruvate kinase 2.7.1.40. For hydrolases, alkaline phosphatase would be 3.1.3.1, acid phosphatase 3.1.3.2, alpha amylase 3.2.1.1, true or acetylcholinesterase 3.1.1.7, pseudocholinesterase 3.1.1.8, chymotrypsin 3.4.21.1, elastase 1 3.4.21.36. 5-nucleotidase 3.1.3.5, triacylglycerol lipase 3.1.1.3, trypsin 3.4.21.4, angiotensin converting enzyme 3.4.15.1. Examples for lyase would be aldolase 4.1.2.3, for isomerase, triosphosphate isomerase 5.3.1.1, and for ligase, that's glutathione synthase having 6.3.2.3 enzyme commission numbers. Aside from that, there are also enzyme theories which explains how the enzyme and the substrate binds. The first theory is the Emil-Fischer's, or otherwise known as the lock and key theory. It states there that the shape of the key, which is the substrate, must fit into the lock, that is, the enzyme. However, the most commonly and widely accepted theory is the Cochran's or induced fit theory. Why is it more accepted? Because it explains how catalysis actually occurs. There's a conformational change that would happen, which would place stress on the bonds within the substrate. That can explain how bonds would break in order for the products to form. Remember that when there is enzyme-substrate complex, a product is generated. So this makes the induced fit model the more widely accepted model out of the two. Let's also discuss enzyme kinetics. Enzyme kinetics class is a chemical reaction that may occur spontaneously if the free energy or available kinetic energy is higher for the substrate than the product. The reaction then proceeds toward the lower energy if a sufficient number of the reactant molecules possess enough excess energy to break their chemical bonds and collide to form new bonds. The excess energy is then called activation energy, and it is the energy required to raise all molecules in 1 mole of a compound at a certain temperature to the transition state at the peak of the energy barrier. At the transition state, each molecule is equally likely to either participate in product formation or remain an unreacted molecule. Reactants class possessing enough energy to overcome the energy barrier participate in product formation. One way to provide more energy for a reaction is also to increase the temperature and thus increase intermolecular collisions. However, this does not normally occur physiologically. Enzymes catalyze physiologic reactions by lowering the activation energy level that the reactants or the substrates must reach for the reaction to occur. The reaction may then occur more readily to a state of equilibrium in which there is no net forward or reverse reaction, even though the equilibrium constant of the reaction is not altered. The extent to which the reaction progresses depends on the number of substrate molecules that pass by the energy barrier. You can see from the illustration above the energy versus the progression of the reaction or the reaction coordinate, indicating the energy barrier that the substrate must surpass to react with and without enzyme catalysis. The enzyme considerably reduces the free energy needed to activate the reaction. The general relationship among the enzyme, the substrate, and the product may be presented as follows. The enzyme first, to be added with the substrate, then there will be enzyme-substrate complex, and then the enzyme and the substrate will break through or under stress, releasing products, and then the enzyme back to its inactive or free state. Different enzymes are specific to substrates in different extents or respects. Certain enzymes exhibit absolute specificity, meaning that the enzyme combines with only one substrate and catalyzes only the one corresponding the reaction. Other enzymes are group specific because they combine with all substrates containing a particular chemical group, such as a phosphate ester. Still, other enzymes are specific to chemical bonds and thereby exhibit bond specificity. There's absolute, group, and bond specificity. There is also stereoisometric specificity. This refers to enzymes that predominantly combine with only one optical isomer of a certain compound. In addition, an enzyme may bind more than one molecule of substrate, and this may occur in a cooperative fashion. Binding of one substrate molecule, therefore, may facilitate binding of additional substrate molecules. Under enzyme kinetics, there are actually two types of reactions. That's zero-order reaction and the first-order reaction. Brief recap. Two of the major influences in enzymatic reactions would be substrate and enzyme concentration. According to Michaelis and Menten, remember class, they hypothesized the role of substrate concentration in the formation of the enzyme substrate complexes. And according to their hypothesis, the substrate readily binds to the free or the inactive enzyme at a low substrate concentration. With the amount of enzyme exceeding the amount of substrate, the reaction rate steadily increases as more substrate will be added. This type of reaction class is following the first order kinetics because the reaction rate is directly proportional to the substrate concentration. Eventually, however, the substrate concentration is high enough to saturate all the available enzyme and the reaction velocity reaches its maximum. When the product is formed, the resultant free enzyme immediately combines with excess free substrate. The reaction in this manner is already in zero-order kinetics, and the reaction rate depends only on enzyme concentration. Okay, you might be thinking, Miss, what are those letters represent? The Michaelis-Menten constant class that is represented by capital K and a very small m. It's derived from the theory of Michaelis and Menten, which is a constant for a specific enzyme and substrate under defined reaction conditions and is an expression of the relationship between the velocity of an enzymatic reaction and substrate concentration. The assumptions are made that equilibrium among enzyme, the substrate, the enzyme-substrate complex, and the product is established rapidly, and that the enzyme plus the product would equate to enzyme-substrate reaction, which is negligible. The rate-limiting step is the formation of product and enzyme from the enzyme-substrate complex. Then, maximum velocity is fixed, and the reaction rate is a function of only the enzyme concentration. As designated in Model 1, you can see there that it is specifically the substrate concentration at which the enzyme yields half the possible maximum velocity. Therefore, the Km indicates the amount of substrate needed for a particular enzymatic reaction to occur. The Km class stands for what again? The Michaelis-Menten constant. The Michaelis-Menten hypothesis of the relationship between reaction velocity and substrate concentration can be represented mathematically as the following, where V stands for the measured velocity of reaction, Vmax is the maximum velocity, while S there is the substrate concentration. The same goes for Km. You will never forget that's Michaelis-Menten constant of enzyme for a specific substrate. Theoretically, Vmax and then Km could be determined from the plot that you can see from this picture. However, Vmax is difficult to determine from the hyperbolic plot and often not actually achieved in enzymatic reactions because enzymes may not function optimally in the presence of excessive substrate. A more accurate and convenient determination of Vmax and Km may be made through a Lineweaver-Perk plot, which is a double reciprocal plot of the Michaelis-Menten constants, which yields a straight line. The reciprocal is taken of both the substrate concentration and the velocity of an enzymatic reaction. The equation for Lineweaver-Perk plot is as follows. Take a look at Model 2, the Lineweaver-Perk transformation of Michaelis-Menten curve. The maximum velocity there, which is represented by Vmax, is the reciprocal of the x-intercept of the straight line. The Km is the negative reciprocal of the x-intercept of the same line. In order to measure the extent of enzymatic reactions, there are actually two general methods that may be used. Number one would be fixed time, where the reactants are combined. The reaction proceeds for a designated time. The reaction is then stopped and measurement is made. While for continuous monitoring or the kinetic assay, from the name itself, continuous monitoring, we will be taking multiple measurements of absorbance changed that are made during the reaction. This type of assay is more advantageous than the fixed time. I am showing you now a table. It's a brief summary of the differences between the endpoint reaction or the fixed time measurement and also the continuous monitoring or the kinetic assay. It says here that in the kinetic reaction, the analysis is determined between the differences of absorbance between 2 points of the progression of the reaction. While for fixed time or endpoint reaction, we determine the total amount of analytes consumed during the progression of a reaction. Usually, class, for kinetic measurements, the reaction is 20 seconds to 1 minute, while for endpoint reaction, the endpoint will be about 5 to 15 minutes. Enzymes class are measured in terms of the change in the substrate concentration, the change in the product concentration, and also the change in coenzyme concentration. When enzymes are quantified relative to their activity rather than a direct measurement of concentration, the units used to report enzyme levels are called activity units. The definition for the activity unit must consider variables that may alter results such as pH, temperature, and substrate. Historically, class-specific method developers frequently establish their own units for reporting results and often name the units after themselves. To standardize the system of reporting quantitative results, the Enzyme Commission defined the international unit, IU or U, as the amount of enzyme that will catalyze the reaction of 1 micromole of substrate per minute under specified conditions of temperature, pH, substrates, and activators. Since specified conditions may vary among laboratories, reference values are still often laboratory-specific. Enzyme concentration is usually expressed in units per liter, that's IU per L, and the unit of enzyme activity recognized by the International System of Units is the Catal units, that's 1 mole per unit for substrate concentration, and the unit of time will be second. So that's 1 mole of substrate per second. These are the following causes of increased serum enzyme levels. Number 1 would be impaired removal of enzyme from plasma. If this happens, class, impaired removal of enzyme from the plasma indicates certain chronic disorders. Another cause would be tissue necrosis and degeneration. Necrotic tissue class is a medical condition in which There are already dead cells in the body organ. The death of these cells happens due to the lack of oxygen and interrupted blood supply. And so, it causes the cells to be acidic, releasing enzymes that break the cells. Third cause would be the increased permeability of cell membrane. And lastly, the increase in the number of cells or the production of cells. Before we end part 1 of clinical enzymology, I want you to write this down. An enzyme accelerates the rate of reaction, thereby reducing the time required to reach equilibrium. A constant change in absorbance per unit time occurs only when the rate of reaction is zero order. A substrate concentration of more than 99 km is needed to achieve zero order reaction. It is easier to measure small increases in product than to measure small decreases in a large amount of substrate. An enzyme do not alter the free energy or direction of a reaction, but it alters the energy of activation by forming a metastable intermediate, the enzyme-substrate complex. Most enzymes are measured by monitoring the rate of absorbance change, so that's kinetic assay, at 340 nanometers as NADH is reduced or consumed, and it allows direct reporting either by IU or KU, that's International Unit, or Catal unit. In first-order reaction, the enzymes are used as reagents to measure a specific analyte. Non-kinetic assay, that is, absorbance is made at 10-second intervals for 100 seconds. Endpoint measurement also determines the concentration of substrate or product at a specific time after addition of the sample. An example of this would be bedside glucose testing using strips. Enzyme activity measurements may not be accurate if the enzyme inhibitors are present. Essential cofactors are not included in the assay and improper specimen storage. Thank you so much for listening, class. That concludes part 1 of clinical enzymology. Wait for part 2 where we will be discussing the major enzymes that has a clinical significance. God bless us all, and please don't forget to take down notes and make sure to study well before the examination. God bless.